Got cancer? Apparently, scientists figured out how to starve it, according to Dr. Ben Miles. Now, this is a video entitled exactly what I just said. Scientists just discovered how to starve cancer. I was sent this video by a friend of mine, and I will admit that I've watched the entire thing through because this person just wanted my own personal thoughts directly sent to him. But it was so good that I decided to actually react to it. So really, if you want to figure out how to starve cancer, if you are someone that is afraid of developing it in some way, you know, whoa, what if I develop cancer and I don't know how to deal with it? First of all, I wrote about this in my book, Contraindicated. If you haven't bought that already, second edition is out now, self-narrated audiobook, paperback, hardcover, all linked in the description below. And there's some pretty conducive methods, especially when you look at how cancer actually works and operates. But if you also have family members maybe that have cancer as well, you can maybe, you know, send this video, my reaction to this video, to them, because this is what we're going to find out all about today. We're going to get directly into this now and waste no further time. Let's get into it.
A portion of this video is sponsored by Radiocode. For centuries, cancer has been thought of as an insidious disease.
What is an insidious disease? That's correct.
Force consuming the body from the inside out.
Yes, effectively, yeah, that's exactly what it does. Cancer cells are deranged cells with deranged mitochondria. The mitochondria are still alive, they still respire, but they can't perform processes like apoptosis to kill the cell when it becomes deranged. It's an unhinged cell. It's on the run, so to speak. It's not constrained. It is unbridled. Yeah, and they solely depend on glucose and all that stuff, so yeah.
1600s, physicians even tried feeding it, literally, applying raw meat to tumour areas in a desperate hope that cancer would select steak over patient. With morbid fascination, the idea of somehow controlling cancer's hunger has permeated everything from old wives' tales recommending fasting.
I feel like a new man.
To their modern
you can't say that fasting is an old wives' tale. It's not an old wives' tale.
equivalent, your friendly neighbourhood wellness guru.
And it was a so-called wellness warrior.
Mel Gibson admits she made up the whole story.
But is there any meat to this idea? Well, maybe, and it comes from a kind of unexpected place. Fat. Those extra holiday pounds may have inspired a team at the University of California, San Francisco, who just published their latest findings in Nature Biotechnology on how fat may be our first weapon capable of starving cancer.
Okay, interesting. Again, I've watched this, so I already know what this is going to be about. In a way, I actually don't like that, because I like extemporaneously reacting to these, because it makes things more informative for you. But anyway.
No chemo, just fat doing what fat does best. Welcome to a series that I'm calling On the Shoulders of Giants, because to me, when you want to peer into the distance and understand the future of what is possible, you need to look down sometimes to see whose shoulders you're actually standing on. So let's cut the fat, reach for another
Okay, humility, good start.
Dorito, and start at the beginning.
Reach for another Dorito? No! Those are contraindicated for human consumption.
How do we understand cancer's hunger for the very first time?
Well, cancer cells, since they have deranged mitochondria, cannot use fuels that are exclusively used to create energy in the mitochondria. So things like fatty acids and things like ketones, they can't use them. So what's left? Well, you've got alcohol, and you've also got glucose. Cancer cells thrive on glucose. Now, from that logic alone, I know that people are going to think that was a sardonic sort of comment, which it sort of was. But just from that logic alone, you can't say that glucose causes cancer, for example. It just means that when you do have cancer, it's really hungry for glucose. So.
In the early 1900s, German physiologist Otto Warburg was...
Yes, the Warburg effect is what he founded, basically, which is exactly what I just described. Cancer cells thrive on glucose. It's the entire reason why they are as hungry of a cell as they are. Because if you were to use glucose to create energy, if you have functional mitochondria, per every glucose molecule, you create 38 net ATP. 38. However, when you can't use that glucose through the mitochondria, so to speak, it's a little more complicated than that. You can only produce 2 ATP per glucose molecule, which means that if it's only depending on the cytosol, the cell fluid, to create energy, or to create ATP really, it must use massive amounts of glucose. So, anyway.
By how cells generate energy. In particular, the differences between normal and cancerous cells and what allowed cancer to grow so quickly. At the time, scientists knew that cells needed glucose and oxygen to survive, but
well, some cells require glucose, okay? Muscle cells require glucose through glycogenolysis. Astrocytes in the brain require glucose through glycogenolysis. Liver cells require glucose not for themselves, but they do require a storage of glycogen to perform glycogenolysis to supply other tissues with glucose, like muscle cells and astrocytes in the brain, and also red blood cells known as erythrocytes through glycolysis. They do the same thing that cancerous cells do, where they use glucose molecules to create 2-net ATP because they don't even have mitochondria. And so they require lots of glucose. Lots. Your red blood cells require lots of glucose. Okay. For every other cell, they do not require glucose and oxygen. That was something that was perceived to be the case back then. You require fatty acids, ketones, and or glucose and oxygen. You're right to say that every cell requires oxygen because every cell has mitochondria, which require oxygen. But that is not the case for glucose. False.
Process like glycolysis, the Krebs cycle, any of those other things that haunt us from high school biology class was still reasonably mysterious. Warburg set out to measure how much oxygen cancer cells consumed. In a petri dish, he laid healthy cells and
I will admit they were not only still mysterious back then. People still fail to truly understand the Krebs cycle even today, in terms of the fact that there are two ways in which the Krebs cycle can actually operate. It's not always an 8-step process, it can be 5 over and over and over again, depending on what substrate the cell is using.
Of tumor in a carefully prepared nutrient solution and connected them to his custom-built manometer to track their oxygen use. The results stunned him. Healthy cells consumed oxygen at a rate he had seen time and time again, but the tumor cells barely used any oxygen at all.
Exactly. So they were still using some, which is why the mitochondria are alive. They're using very little, though. That's the problem.
Believing this was maybe a mistake, potentially due to lack of nutrients in the solution, he topped up the glucose levels in the petri dishes and ran the experiment again. This time, the results were even more surprising. In the presence of excess glucose, cancer cells stopped using any oxygen at all. But how could this be? Confused, Warburg began adding various testing agents to his...
Well, by the way, there's your sign that maybe eliminating glucose from the diet can help with the sort of rekindling of mitochondria. However, there is nuance behind this, and we will cover that in this video.
Solution. When he added phenol red, he saw an immediate bright yellow hue fill the petri dish. His fast-growing cancer cells were thriving in a pool of acid. Chemical analysis revealed it to be lactic acid, the compound also produced by muscle cells under extreme exertion. But this wasn't just a minor
Well, no, they're produced by muscle cells at all times. Right now, I'm producing lactate, not lactic acid, because under physiological pH, in cellular pH and also arterial and venous pH, it will exist as lactate always. But anyway, they're producing lactate right now because when a cell uses glucose through glycogenolysis, the byproduct of that is pyruvate, and it builds up a lot, and so then what happens is you produce lactate. Pyruvate is reduced to lactate. That's what's going on. The lactate is then exported into the bloodstream, migrates to the liver to be converted back into glucose, which is exported into the bloodstream, sent back, and then picked up by that muscle fiber again for the next twitch. It's amazing how the body works. It's a cycle. It's a circle. Cori cycle is what it's called. So, yeah. Anyway, that is why this cell was producing lactate. It's because it's a byproduct of glucose metabolism.
In some cases, Warburg recorded up to a 70-fold rise in lactate production compared to normal cells. Then came the real revelation. He measured glucose uptake across both cell types. Normal tissue consumed about 16 milligrams of glucose per 100 cc of medium. Tumors, over 70 milligrams, a 4-fold increase. Warburg, for the very first time, had proven that cancer was genuinely a disease of voracious appetite. But the core mystery remained. If cancer cells weren't oxidizing glucose, what...
Okay, so this is the pedant in me, but you can't oxidize glucose. You can oxidize pyruvate, because oxidation requires mitochondria. So I was just talking about how cells without mitochondria, like erythrocytes, they don't have mitochondria. They use glucose to create ATP. Well, they're not oxidizing it because they don't have mitochondria. Okay, they're metabolizing glucose into pyruvate to produce ATP, whatever. That's what's going on. You don't oxidize glucose, you oxidize pyruvate. Pyruvate can be oxidized if you have mitochondria in the cell. If not, it's converted to lactate, and then just like the cancer cell's doing.
In healthy cells, glucose is broken down through glycolysis, then shuttled into the mitochondria,
Powerhouse of the cell.
for oxidative phosphorylation, a highly...
Well, pyruvate is ushered into the mitochondria if your cells are propitious towards using pyruvate for oxidative purposes. But in my body, for example, since I don't consume glucose, that pyruvate will probably be converted into lactate because it's going to be blocked out of the mitochondria because my body's using fat instead. It's a Randall cycle situation.
Process that for each molecule of glucose produces over 30 molecules of ATP,
38 specifically.
energy used by cells. But in cancer cells, Warburg saw something else entirely, a reversion to a primitive, inefficient pathway. They were stopping at glycolysis, producing just 2 ATP per glucose.
Yes, because, yeah, glycolysis only produces 2 net ATP. Pyruvate entering the mitochondria is the transition stage, or the preparatory phase, is what they call it. It's actually separate from glycolysis. That's why when people say aerobic glycolysis, that's not a thing. Glycolysis is strictly anaerobic.
Dumping the rest as lactic acid. Warburg proposed
Lactate.
something radical, that this switch wasn't a symptom of cancer, it was the cause. Warburg believed that damaged respiration, a defect in the mitochondrial function, forced cells to adopt glycolysis as default, and that this shift was what triggered uncontrolled cell growth.
Exactly, which is still the prevailing theory. And basically what that means is that cancer is a mitochondrial dysfunction issue. It is a metabolic dysfunction issue, and it's really characterized by a derangement of mitochondrial DNA. You have DNA in a cell in the nucleus of the cell, but you also have mitochondrial DNA that's separate. And you derive your mitochondria from your mother, by the way. So you have the same genetic sort of lineage there. So anyway.
Idea would become known as the Warburg effect, that cancer would throw efficiency to the wind and take a growth at all costs approach. While not completely universal, this increased glucose consumption is seen in 70 to 80% of cancers to varying degrees. We can actually see cancer's hunger in real time thanks to a technique called positron emission tomography, or PET imaging. It works by injecting a patient with radioactively labeled glucose, essentially glowing sugar, then tracing where in the body that glucose gets consumed most rapidly. That bright spot in the pancreas is a tumor, so metabolically active that it's comparable with the brain and heart in terms of glucose usage. It's not just growing.
Yeah, so once again though, I want to say something about this. So comparable to the brain and heart in terms of glucose usage. According to whom, on the basis of what? The reason I say this is because this is based on a population of people that are consuming carbohydrates primarily. If you don't consume primarily carbohydrates, your brain is not using that much glucose. It's still using some. It's still using far more than most of your other organs. Absolutely. Absolutely. But it is not using glucose nearly to the same degree that people that are consuming carbohydrates regularly are. So that's something to consider here. And same thing with the heart. The heart primarily uses fatty acids, actually. And it relies on ketones, it seems, during heart failure. Anyway, I want to put that in there because I don't like whether it's subconscious, unconscious, or conscious. It doesn't matter. It doesn't matter. These sort of tacit insertions of things, these side comments, they put images in people's heads, whether they realize it or not. The brain does require glucose, but the neurons of the brain do not. No, they don't.
It's feasting. In this second image, taken after several rounds of chemotherapy, that bright spot is gone. The tumor's metabolic signal has vanished, hopefully a sign that the treatment has worked. Up until his death, Warburg was convinced of just one thing, that cancer's hunger could be its greatest weakness, and that became a very compelling idea. Could the treatment for cancer be as simple as just limiting its energy supply? What?
Well, yes, but the difficulty is actually, this is where the nuance comes in. The difficulty is actually finding a way to limit its glucose supply. Why do I say that? Okay, it's a superficial argument to say, okay, you can remove glucose from your diet and then you'll have an amelioration of cancer. I actually wrote about this in my book. Again, buy that if you haven't already, because, I knew about this before this video was out. The problem, though, with that is believing that the body will not upregulate its production of its own glucose just to subserve the needs of the cell. Because here's the thing, a cancer cell is not exactly a foreign cell. The body still recognizes it as its own cells. The mitochondria are dysfunctional, sure, but it's still a cell of the body. So if the cell is in need of glucose, the body will create more glucose to upregulate glucose production and provide the cancer cell with glucose. At least that's a theory. And you're going to see later on that it seems to be that's the reason. That's what happens. So let's see what he has to say, and I have some comments about that as well.
That question, but first I have to thank the most relevant sponsor possible, Radioco.
Okay, why you can't starve cancer or all your nutrients belong to us. Okay, whatever.
Although research didn't just influence science, it lit a spark in public imagination. The idea that you could starve cancer by cutting off its fuel supply became deeply appealing, especially to those seeking alternatives to mainstream medicine. The earliest approaches were surprisingly straightforward.
Okay, so all the food that is appearing on this screen right now is contraindicated for human consumption, okay? Plants you should not be consuming, carbohydrates you should not be consuming, fruit, which is a plant, at least it's the fruit of a plant, don't be consuming that either. We should be consuming a species-appropriate, species-specific diet, or a diet that best approximates that diet based on every resource we have in the modern world and based on every inference we have about the ancestral world, which is a diet consisting primarily of the flesh and associated fat of large ruminant animals, with added fat in the forms of butter, tallow, lard, suet, and ghee according to your appetite, so to taste, salt to taste, and water, which is inferred by many different things, but this video is specifically about cancer, so I'm going to be a little sparing here.
Cancer cells thrive on glucose. Could removing sugar and carbs from the diet cut off their supply? Enter the ketogenic diet. Originally developed in the 1920s to treat epilepsy, keto...
Well, no, the ketogenic diet was not invented. It was discovered millions of years ago and was embarked upon and consumed for millions of years of our existence, if you include proto-humans that preceded our current speciation, that being Homo sapiens sapien, and only until very recently were we consuming any significant degree of carbohydrates at all. Our efficiency for gluconeogenesis in our bodies is that of hypercarnivores, which tells us that, one, there was a positive selection pressure that was imposed upon us for thousands, tens of thousands, hundreds of thousands of years to even be able to produce our own glucose, which tells us we weren't consuming glucose to any significant degree. And also, considering we still have that efficiency, plus or minus a very minimal variation ostensibly, that there wasn't a negative selection pressure to knock that efficiency level down. So we still should not be consuming carbohydrates at all. The exact required amount, therefore, due to our efficiency, is 0. It's that simple, and above a certain physiological concentration in the bloodstream that is most conducively achieved by consuming it is toxic, evinced by diabetes, chronically elevated blood glucose, and nothing else, explicitly defined and measured as such. So, no, it wasn't invented in the 1920s. It was rediscovered, in a way. There were still plenty of things that are considered keto that we still were not consuming. Like leafy greens are keto, for example. Shouldn't be consuming those either.
New life among cancer diet advocates. By eliminating carbohydrates and replacing them with fats and proteins, the theory went you could lower blood glucose and starve the tumor. On the surface, it sounds...
Well, it's not necessarily about lowering blood glucose per se. I mean, it's lowering it more, sure, but I mean, of course, it's not going to cause you to be hypoglycemic or something, or ketoacidotic. Anyway, but this is the point. Your body's going to get the glucose from somewhere. Gluconeogenesis is right there. So this is why there's ambiguity here.
Entirely scientifically possible, but trial after trial failed to show any consistent benefit.
Okay, so I would like to see the trials. However, I'm not extremely passionate about it because I just explained why this could very well not be the case. I wrote about this in my book, that one of the protocols I recommend is cutting out the glucose because it's really not, if you have cancer, it's not doing you any favors. And the theory goes that, okay, you could starve cancer cells. And the theory still holds, actually, and I'm going to explain it. He's going to explain it as if it was disproven. And I would be totally fine accepting that it was disproven, because the idea is really to not develop cancer at all in the first place. That's the goal, right? But even after this video is over, there's a contradiction here, and I'll explain it at the end.
Not for tumor shrinkage, not for survival, and that wasn't unique to keto. Every so-called anti-cancer diet followed the same pattern. Big promises, disappointing results. One of the most prominent examples, the Budwig diet, was deeply influenced by Warburg's theories. Dr. Johanna Budwig believed cancer was caused by faulty cellular respiration, specifically that cells weren't absorbing oxygen properly due to a lack of essential fatty acids. Her solution...
Okay, so interesting theory, because there is a lack of oxygen uptake in cancer cells, as we already covered, but the mechanism doesn't really seem to make sense. The blood provides the body with oxygen, the cells with oxygen, and the mitochondria take it up, unless there is a problem with their function that is ostensibly caused by DNA derangement, which is due to externalities that the body is not genotypically evolved to withstand. So there you go.
Blend of flaxseed oil and cottage cheese, equal parts delicious as entirely unlikely to work. She claimed it could restore healthy fat metabolism and oxygen uptake in cells, but while her recipe may have made headlines, no scientific study ever showed it could impact cancer's outcomes.
Well, to be fair, doctor, there are no studies in the area of human nutrition science that can show an impact of anything, because impact is a cause and effect statement, and no studies can inform upon causality in the area of inferential statistics, because inferential statistics makes no claims about causality, precisely because inferential statistics cannot involve control of your cases. It can't control for confounding variables. It's not possible. So let's be consistent here.
Was wishful thinking wrapped in scientific language. The alkaline diet went down a similar path.
Yeah, the alkaline diet was absolute nonsense. The entire philosophy behind the alkaline diet was cancer cells are acidic because of the fact that they have lactate buildup, right? Oh, the cancer cells are acidic, so now you need to reduce your acidity in your diet. Yeah, in the body, cancerous cells are slightly more acidic because of lactate buildup. Sure, fine, but they cannot go below a certain pH level or else the cell will die. In order to preserve the pH balance within the ranges that are physiologically appropriate, the lactate that builds up will be exported, puff, out of the cell. That's what happens. That's what happens. It's ridiculous. You cannot influence the pH of your solutions in your body with any meaning whatsoever. It is tightly controlled. The only thing that you can do is influence the acidity level of your urine and your stomach, the latter of which is very transient because it adjusts very quickly.
That certain foods could shift the body's pH and neutralize the lactic acid buildup around tumors, but it mistook something fundamental, that the acidity is a byproduct of cancer metabolism.
Exactly. It is not the cause of it. That was a very... Yes, I already covered this, a year and a half ago now?
Not the cause. And more importantly, the body tightly regulates pH, regardless
Yes.
what you eat. Again, no evidence of benefit. And then we got into slightly more dangerous examples like Belle Gibson's whole pantry movement. She built an entire wellness empire on the claim that she'd cured her cancer with diet and lifestyle changes. But in the end, the only whole thing about it was how much she'd made up. She later admitted she'd never had cancer to begin with. Time and time again, these miracle diets failed.
Wow.
Test of scientific evidence. Some were rooted in genuine scientific questions, others were pure pseudoscience. But none could outsmart the problem at the heart of cancer. It is exceptionally good at finding what it needs.
Yes. This is the problem with cancer. That's the problem.
The tumor grows, it burns through nutrients faster than its surroundings can provide. The local tissue becomes hypoxic, oxygen starved, and the tumor responds by sending out molecular distress signals, VEGF, or vascular endothelial growth factor. This triggers nearby blood vessels to sprout new branches, a process called angiogenesis, delivering fresh blood, oxygen, and nutrients straight to the tumor's door. And this brings us to the brutal truth. This is the fundamental flaw in any whole body approach to starving cancer, whether through diet, fasting, or anything else. Even if you put your entire body into a nutrient or energy deficit, cancer cells have...
You cannot go into an energy deficit. And also we're not talking about nutrient deficit either, and with respect to micronutrients. Nor macronutrients actually, because a carbohydrate deficit doesn't exist in terms of exogenous inputs. The exogenous requirement for carbohydrates is 0 across the board. Okay? With very small exceptions that haven't even been proven to be exceptions. It's a theory that I've put forth.
To protect themselves, they suffer less than the surrounding healthy tissues, or put more simply, you starve before the cancer does. And to be clear, this isn't just a problem for alternative medicine, this is a challenge even for traditional clinically tested therapies. One of the most accidentally fascinating discoveries in this space come from a drug you've probably heard of, metformin. Prescribed to lower blood glucose in diabetics, metformin...
Yeah, it's prescribed to cause the diabetic patient's cells to pull glucose from the blood by inhibiting the electron transport chain of their mitochondria. It inhibits complex 1. It starves the cells of energy, particularly if you're consuming glucose though, because it only inhibits complex 1. Most electrons, when you're consuming fat, they go through complex 2, but some still go through complex 1. So anyway.
One of the most widely used drugs in the world. Over 86 million prescriptions were filled in the US alone in 2022. When researchers looked at cancer rates among diabetic patients, they noticed something striking. Those taking metformin were significantly less likely to develop cancer.
Not less likely. There was a lower incidence rate observed between the two groups. Less likely, or likelihood, indicates probability, which cannot be informed upon in this area of science, despite the fact that it is based on inferential statistics, which claims to be able to prove probability. But no, you can only inform upon the likelihood of something if both of your groups have all confounding variables controlled for.
Anywhere from 30 to 50% compared to diabetics who weren't on the drug. But if a patient went on to develop cancer, that benefit disappeared. Metformin didn't improve survival rates, it couldn't stop a tumour that was already entrenched. So the question became, if cutting supplies off at the whole body level doesn't work, what if we flipped the problem around? What if instead of starving the entire system, we robbed the tumour right at its doorstep, just before the nutrients ever arrived? In 2002, a group of radiologists and researchers split between the University Hospital Zurich and the University of Ottawa made an unexpected observation. While analysing PET scans, they noticed a consistent, strong signal lighting up around the neck and spine in some adults. At first they assumed it was just muscle activity, which does uptake more glucose when active, but something didn't...
Yes, it uptakes more glucose the more active it is, but it is always uptaking glucose. In fact, you require glycogen for a muscle fiber to twitch in the first place. Even ones like this.
The signal got even stronger when the subjects were exposed to cold temperatures. Curious, they compared the PET
There's your sign.
the CT scans and realized this tissue wasn't muscle at all. It was brown adipose tissue.
Yes, which is different from white adipose tissue, particularly because it has more mitochondria that are uncoupled, meaning that the proton gradient is uncoupled from ATP synthase. It's a very interesting phenomenon, and it happens actually in ketogenic states under times of low insulin. Just want to throw that out there.
Also known as brown fat. Brown fat is very different from white fat that most of us are familiar with, the kind that simply stores energy.
Yeah, so basically brown fat, what it does, it can get into the biochemical granularities of it, the differences, the distinctions between white and brown fat. However, that would be gratuitous. Basically, the major difference is that brown fat will take up more nutrition, more substrate, so fat, glucose, whatever it may be, to create the energy that it requires than it actually requires. So basically, you have an energy demand from a white adipose tissue. Usually what happens, what a cell does, is it takes just as much substrate as is required to create the energy that it needs. Brown adipose tissue take up way more than it actually needs, and it expends the rest of it as heat, okay? So brown adipose tissue is designed to basically heat the body up. It's non-shivering thermogenesis, basically. That's what we're talking about here. Babies have lots of brown adipose tissue precisely because they cannot shiver like human beings do, or like human beings do, like adults do, sorry. Babies aren't humans. Anyway, it's typically believed that you lose a lot of your brown adipose tissue the older you get, but you don't necessarily have to. You really don't. It's kind of hard to turn white fat into pure brown fat, I think, but you can definitely brown it. Like you can turn it into beige fat. But I believe that you can turn it into brown adipose tissue, frankly, if you do. Anyway, the reason for the distinction in color is because brown adipose tissue have a lot more mitochondria in them, and mitochondria have a lot of iron in them. That's your reasoning.
Fat burns energy. It generates...
It doesn't burn anything. It oxidizes things, and it also doesn't oxidize energy. It oxidizes substrate, mass.
Keeping the body's temperature stable and is critical for newborns who can't shiver effectively yet.
Yes, there you go.
Scientific consensus at the time was that brown fat virtually disappeared after infancy.
Yeah, the only reason that was purported to be the case was because everyone started consuming carbs and, turned their fat all into white fat that's just storing fat. That's what it was. That's it.
Weren't supposed to have it, yet there it was, glowing brightly in the scans. Subsequent studies, particularly by a group of researchers in Japan, confirmed it. When healthy adults were kept in a 19 degrees Celsius room,
This is the room.
cool enough to trigger thermogenesis, but not cold enough to cause shivering, their brown fat lit up on PET scans. Not only was it still present, it was metabolically active.
Take some cold showers. I've been doing that recently. I'm not used to it yet. I've never done this in my life, but I'm getting used to it.
At the Karolinska Institute in Sweden, researchers decided to take this discovery one step further. They wanted to understand how brown fat might interact with cancer. They designed a simple experiment. Mice were implanted with tumor cells and then...
Okay, so mice. I understand why you had to use mice, but it does... Just because the limitation is understandable as to why it is a limitation doesn't mean that it is not a limitation. That is a massive limitation here.
into two groups. One group was kept cozy, living at 30 degrees Celsius, a temperature that's warm enough that the body doesn't need to generate extra heat. The other group was moved to a brisk 4 degrees Celsius environment, cold enough to activate thermogenesis, but not cold enough to cause constant shivering. Over the next several weeks, the researchers carefully tracked tumor growth and glucose uptake using PET scans, and the results were compelling. In the warm environment, tumors showed strong uptake of radioactive glucose, just as you'd expect, but in the cold exposed mice, it was a different story. The brown fat activated for thermogenesis, competing aggressively for glucose, and as a result, the tumors became starved of their primary fuel.
Okay, so this is where I want to pause this. The implication here is that the brown adipose tissues, since they're a lot more energy hungry, so to speak, they kind of meet the energy hunger that cancer cells have, so they compete for glucose from the same source. Imagine if you have a hose, so think of the hose as your blood vessels pumping blood through. The water inside is the blood, okay? Now imagine that hose that you have is attached to a pipe with one end. You're going to have water spilling only out into that end, right? That's it. Now imagine that you take that attachment off and you put one on that branches out into, think of the shape of the goalposts on a football field, American football for my international viewers, and you've got two hoses or two entries, or rather exits for water. Well, what's going to happen is that the water flow, the water pressure in both of those is going to be diluted. And that's sort of what they're saying. They're saying, well, if you have brown adipose tissue, it pulls more. It has another pressure pulling glucose from the blood. My problem with this philosophy that starves, or that helps to starve cancer, is, well, the cancer cell would therefore be losing some glucose, so why wouldn't it just ramp up gluconeogenesis even more? Because imagine, in the hose analogy, if you turn up the water pressure even more, well, then the pressure's still going to increase in both sides, despite the fact that it's less in each side than it would be if it were just one exit, like before. But it doesn't matter. This is why there's some ambiguity here. If you're going to say that this works, why can't you say that carbohydrate restriction works? There is something odd happening. This is what leads me to believe that I need to look at the trials that were used, that he was referencing before in the ketogenic approach, to see how they were done. I mean, really, how was it done? I need to look at the demographic. I need to look at the sample size, the length of the study, all that stuff. It is weird. It is odd. I'm not saying he's wrong. I just think there's a discrepancy here. The mechanism is unclear. It seems contradictory. This is why I wanted to make a video about this.
Of which was dramatic. Tumor glucose uptake dropped significantly. By day 20 after tumor implantation, researchers observed an 80% inhibition of tumor growth compared to the warm group. And
I mean, the thing is if it works, the mechanism is just very unclear. Why are we able to say, according to this mechanism, that carbohydrate restriction wouldn't work? Seems like it would also do the same thing. Here's the great thing, though. Here's the ironic thing, as I just covered. If you embark upon a ketogenic diet, your insulin is lower. You can turn your white fat. White fat starts to brown under times of low insulin. So carbohydrate restriction also is still conducive, even according to this logic, to ameliorating cancer, apparently, ostensibly. I mean, come on. Just maybe not for the reasons that we're saying. Not because we're reducing glucose supply, but because we're reducing insulin, which uncouples white fat, turning it into brown fat, thus starving cancer cells. So it still starves cancer? This is what I'm saying, guys. This is odd. This is very odd, okay? So yeah, it would be great if you could turn your white fat into brown fat. Oh, wait, you can. Stop consuming the very macronutrient that isn't really a nutrient to begin with, exogenously speaking, that being carbohydrates.
more striking, the overall survival rates of cold-exposed mice was double that of the warm group. But could this cold-induced effect actually work in humans? In 2021,
That is the question.
researchers tested the idea in a very limited but intriguing human study. They worked with a patient diagnosed with Hodgkin's lymphoma, exposing them to mild cold, just 16 degrees Celsius for 4 days. PET scans showed clear activation of brown fat, and more importantly, a noticeable decrease in glucose uptake at the tumor sites. It wasn't a cure and it wasn't comprehensive, but it was the first real evidence in a human that brown fat could outcompete cancer for its fuel, effectively starving a tumor. A glimmer of hope, maybe just maybe, Warburg's century-old findings could still shape modern cancer therapy. But there was obviously a problem. To maintain the effect, the patient would likely have to stand in what's essentially a walk-in fridge for weeks, possibly months.
Hey, look, a freezer man!
While also avoiding sugar or any excess glucose. Not a perfect recipe for a body already weakened by battling cancer.
Well, the sugar thing absolutely is. Yes, it is. Fact.
The question became, could we take these ideas and turn them into something practical, something targeted, controlled, and therapeutic?
Well, reduce carbohydrate consumption to 0, and also, I guess, according to this, maybe do some cold therapy, like taking cold showers or doing ice baths or something. You want to gradually do the ice bath thing, because, I mean, you can go into shock if you do it too quickly. The cold showers, though, that's a good introduction.
Something that didn't require living in a freezer.
Well, okay.
Look at that blubber fly. Nurse, cancel my 1 o'clock.
Just published in Nature Biotechnology, scientists at the University of California, San Francisco asked a bold question. Could we skip the freezing cold and still harness brown fat's metabolic power? Or more simply, keep the hunger, lose the shivering? Using CRISPR, they began genetically engineering white fat cells to behave more like brown fat. By inserting upregulated specific genes from brown fat cells, they aimed to create a new kind of cell, one that could outcompete cancer for nutrients without needing to be cold activated. They called these hybrid cells the slightly unattractive sounding beige fat. To figure out what genes worked best, it
would be best if we could just transform the white fat into brown fat, which we know we can do. But they're trying to find an intervention to employ that they can profit from. So, I mean, anyway.
Transwell experiment, where 2 different cell populations share the same nutrient pool but are physically separated. One gene in particular stood out, UCP1.
Yes, uncoupling protein 1, exactly, which is present in fat cells and is basically activated under times of low insulin and cold exposure as well, as we already covered. I already knew about the cold exposure thing, turning it into brown fat, white fat from brown fat, but I mainly focus on insulin because I'm focused on diet on this channel.
Key player in thermogenesis. When they tested UCP1 modified fat cells against cancer cells, the results were shocking. At the end of the experiment, almost no cancer cells remained. Meanwhile, the beige fat cells were thriving. Worried this was an error, they repeated the trial again and again and got the same outcome every single time. They had taken the competitive fuel-guzzling metabolism of brown fat, supercharged it, and severed its dependence on cold. From fat, they had made a weapon. To test it in a living system, they turned the most efficient UCP1 modified cells into fat organoids, tiny lab-grown clumps of tissue that function like miniature fat organs, and they implanted them next to tumor sites, like a set of metabolic love handles poised to siphon off nutrients before they ever reached the tumor. 3 weeks later, they compared tumor growth with a control group. Now, fair warning, if you're a little bit squeamish, you might want to look away, but here's what they found. Tumors adjacent to the beige fat organoids has shrunk by more than 50%. And they weren't just beating one type of cancer, they outcompeted aggressive cell lines from breast, pancreatic, colon, and prostate cancer. No chemotherapy, no radiation, simply by being better at consuming glucose. Turning one of the body's own metabolic tools into a precision weapon against one of its greatest flaws. The researchers called it living cell therapy, and fat, it turns out, is a perfect medium for it. Fat cells are easy to extract. We've been doing it through liposuction for decades. They grow well in a lab, can be genetically modified with precision, and crucially, they're easy to re-implant using well-established medical techniques, as evidenced by the myriad of enhanced buttocks that we've seen parading around the earth, not a topic I thought I'd cover in this video. Most importantly, fat plays nicely with the immune system. We know this from decades of cosmetic surgery. Reimplanted fat generally integrates smoothly without triggering serious immune rejection. That makes it an ideal candidate for a cell-based therapeutic. Now, of course, there is still further work to be done.
So this is far better than chemotherapy, obviously, but why wouldn't you just go through a holistic approach to turn your white fat into brown fat instead of getting fat taken from you and then reintroduced into you as a separate organoid, like you said, around the tumor cells? See, this is what I'm talking about. If you need to, then go ahead for some reason, but anyway.
Refining the method, scaling it up, and eventually moving through rigorous safety and efficacy trials. And it's very reasonable to ask questions like, what if the tumors respond by ramping up angiogenesis?
Exactly.
Or if you manage to starve them a little, many cancers might just switch metabolic gears, burning fat rather than glucose.
No, they can't do that. They can't burn fat. They cannot do that because their mitochondria dysfunction. I mean, anyway.
Tackling those problems is maybe for the next generation of scientists to solve, but now at least you know whose shoulders you're standing on. We aren't there yet, but the concept is here, and the biology broadly is sound. So imagine
biology is more sound than inferential statistics, but it is never fully sound.
Perhaps not too far off, where instead of flooding the body with radiation or chemotherapy, we seed it with enhanced cells engineered not to attack the cancer directly, but to starve it. And we turn one of the body's softest tissues into one of its sharpest medical tools.
And we already know how to do that holistically.
Day in the future, cancer may be left out to starve.
Well, there you go.
I often come back to about standing on the shoulders of giants. It's largely how I think about science, and it's the bit that has always been interesting to me. It's the unbroken chain of curiosity, often stretching back across generations of the smartest folk that we have on the planet. Someone asks a question, someone builds a tool, someone else runs an experiment. There's always false starts, but everything seems impossible until someone actually does it. That's the part that's always drawn me in, not just the breakthroughs, but how we actually get there piece by piece, standing on what came before us. I feel super lucky that here on YouTube, as well as in my day job, understanding the future of what is possible and working alongside those scientists to actually make it happen is part of what I get to do. I'm super grateful I get to play a small role and that I get to share a glimpse of those journeys with you guys here. That's the part of this that I love the most. There have been a lot of new folk joining the channel recently, since at least my last long video. I know what you're all actually here for. We have some really cool stuff coming up in the next few months I'm excited to share with you. The algorithm thinks that you might like this video next, so maybe check it out. Thanks very much for watching, and I'll see you guys next time. Goodbye.
I feel like a new man.
All right. Well, I will say right now that I think that this video involved me talking the least that I've ever talked before, because for the most part, everything was good. The mechanism is just a little unclear here when it comes to why carbohydrate restriction can't work, but this can work apparently because it starves the cancer. Anyway, I thought that I would use this video sort of as a way to bring awareness to the fact, though, that brown fat can actually be, you know, a cancer therapy. I thought it was actually a pretty good video, as much as I was like, no, they can't use fat. Okay, that was a little frustrating. You said in the beginning that they can't use fat. I mean, I guess he didn't say that. He said that they just use a lot of glucose, but come on, man, come on. Anyway, yeah, so cold therapy, restricting carbohydrates to 0. There you go. And knowing that this therapy works if you, for some reason, have to get this introduced into you, I mean, it's far better than chemotherapy. Western medicine is not the worst thing in the world. It's just been abused. And the philosophy behind it, a lot of times, is just, it's anti-human. Like our genes are inherently flawed or something like that. Okay. Anyway, but if you enjoyed the video, if you found it informative, leave a like. Subscribe to the channel if you haven't already. If you haven't subscribed to the channel, what are you doing? What are you doing? Leave your thoughts in the comment section below as well. And also, buy my book, Contraindicated. Again, a closer look and revision of mainstream health axioms that have perpetuated illness, disorder, and disease for over a century. If you have not already, subscribe to the Patreon if you haven't already as well, to gain access to one-weekly uploads, ad-free content, uncensored content, and an exclusive Discord server. And finally, a lot of times I am reacting to inane morons, imbecilic, nescient morons, just ignorant people online that are infuriatingly ignorant. And a lot of times they have authority tags next to them, whether it be PhD, MD, or coach. For example, there are a lot of nutrition and health coaches online saying a bunch of very superficial, simplistic things. They don't know how to critically think about issues. And it puts a pressure on people like me, people like you, to stand up against this nonsense. However, if you felt the desire to stand up against this nonsense, but you feel inadequate, you feel like, well, no one's going to listen to me, I've already had the experience of people not listening to me because they expect some sort of authority tag. Fortunately for you, I have a solution to this issue of your absence, your bereftness of an authority tag. And that is with a program called basically Carnivore Certification United. It is a certification program headed by carnivore coach Peter that I am affiliated with that allows you to become internationally accredited or internationally certified as a coach. A lot of people don't realize that if you wanted to become a coach, you could just call yourself a coach legally and there would be no repercussions. It's allowed. It is totally legal. But obviously that's ridiculous. You become accredited or recognized as an official, authority figure that has perks or anything by simply calling yourself a coach. And so we have become internationally certified and accredited, with more on the way, by the way, making us even higher in the sort of veracity hierarchy, so to speak, the authority hierarchy, that allows you, once the program is passed, to become internationally accredited, an internationally certified coach, which allows you to, for example, acquire more insurance to protect yourself more, which every doctor, everyone has. I know that we've been attacked for, you get insurance because you're going to hurt people and harm people's health. Every single doctor has insurance, even the one you trust, okay, for the same reasons. So it is important because everyone makes mistakes, and sometimes those are out of your control. It helps hedge against those issues, but in order to learn more about that, we have the website for the program linked in the description below, as well as Peter's contact information for you to negotiate a plan with him to sign up for the program. I've contributed 4 modules to that program. It's fun. There's a great community. Peter has been coaching. He's very experienced. He's been coaching for over 20 years. If you want to learn more about the program and who he is as a person, I've linked the interview between myself and him in the top right corner of the screen that we did a few months ago, which is very informative if you're still on the fence about it. But with that being said, I look forward to seeing you in that program. Now, join me next time when we're reacting to more inanity, more groundbreaking scientific information, potentially, reviewing newsletters, studies, monologuing about something, venting about something, etc., etc. So, till then.